Okay. Glycolysis. Pretty much there's 10 steps. The goal of glycolysis is to split the glucose molecule into two molecules. The end product is going to be pyruvate. You need pyruvate for the Krebs cycle, so that's kind of like the whole spectrum of things. So we're going to go by it step by step. There's 10 steps. It starts out with a single glucose molecule in step 1. Pretty much what happens is you have this enzyme. It's called hexokinase. It uses one ATP molecule, and that ATP comes in and creates a glucose 6-phosphate. That's the I guess the froth for the second step. So after that, you have one ATP kinase going away. In step 2, you start with that same glucose 6-phosphate. You have like a phosphate attached to it, and it becomes fructose 6-phosphate, which I guess how it becomes is irrelevant. It just becomes fructose, which is important. But you still notice it still says phosphate. You still have that phosphate on it. Then step 3 is when you pretty much do the same thing you did in step 1, where you add an ATP molecule and phosphate, so another phosphate gets attached to it. In this case, it's a phosphofructokinase, which is an enzyme that's used to attach another phosphate. Then what happens is that addition of the ATP, of the actual phosphate, becomes fructose 1,6-biphosphate. So you can pretty much imagine a fructose molecule, and it has two phosphates attached to it. And that's where we'll go to step 4, I think, is where that fructose 1,6-biphosphate, if you can imagine it, it's kind of getting cut right in half. And like long story short, you don't have two of the same molecules. They're isomers of each other. And step 4 is all about making like the one you want. It's called glyceraldehyde 3-phosphate. You get one of those from step 4, and you also get isomer. And you change the isomer into another glyceraldehyde 3-phosphate. We'll go over it. Yeah, sure. Right. So that's the next step, right? So you have, after step 4 is all done and step 5 is all done, you get 2 glyceraldehyde 3-phosphates. And this is really important. We talked about this in the lecture. Anything that happens to one is the same exact thing that happens to the other. So I'll focus mainly on one, but you have to know it's doubled. So you start with this glyceraldehyde 3-phosphate, and you take this NAD plus molecule, and you add a hydrogen to it using this enzyme. It's called triose phosphate dehydrogenase. So you make one NADH. Now keep in mind, on the other side, because there's also another glyceraldehyde 3-phosphate, you are making another NADH. So after that step, it's step 6, you're going to have 2 NADHs. Also during this phase, there's organic phosphate kind of floating around in the cytoplasm. This organic phosphate attaches to this glyceraldehyde 3-phosphate, and yeah, I think that's what happens. Yes, yeah, okay. And you end up with, yeah, that's where you get this bis from in step 7. So you have 2 phosphates on that molecule again, kind of like you had earlier. So you have two phosphates on it. And then what happens, so because you have 2 phosphates on the molecule, what actually happens in step 7 is you make ATP now, which is really, really good. So you take one of those phosphates off. It comes off and makes ATP using this enzyme. It's called phosphoglyceralkinase. So you make one ATP. Now the same thing is true too. You make one ATP over here because there's the same things going on. And then you end up with a molecule called 3-phosphoglycerate. Now it only has one phosphate on it now. So that's step 7. We'll go to step 8. You want to do it, or? You can. All right. So you get from step 7, you get the 3-phosphoglycerate. The phosphate, it's not in an ideal position. It's got to move to a more, I think, unstable position in the molecule, because eventually it's going to come off for ATP. So this phosphoglyceromutase is an enzyme that moves the phosphate to a different place on the
on the molecule. So it moves from the third carbon to the second carbon. And then that molecule then transforms using the enzyme inulase. That reaction, it's dehydrogenase. So two waters come off of it, and that turns into phosphoenol pyruvate, which is the molecule right before pyruvate that we need at the end, but they call it PEP for short. And then finally the last kinase, pyruvate kinase, transforms that phosphoenol pyruvate into pyruvate, which is what we needed for the next step. But in that process, that phosphate that's on there, it is added to ADP to make ATP. So we also get another ATP molecule. And remember, this all happens twice, like Justin said earlier. So we'll end up with 2 ATP here. And we created 2 ATP over there.
And 2 pyruvate, right?
And 2 pyruvate. Yeah, so you made 4 ATP and you used 2 ATP, so you gain 2 ATP in the end.
Yeah.
But that pyruvate then, this is what pyruvate is, that moves into the citric acid cycle or the Krebs cycle, and we don't need to learn all the steps. There's several molecules that it transforms throughout here. But it's a cycle, so it's self-regenerating. So pyruvate comes in here, it turns into acetyl-CoA through several different transformations in this cycle. Carbon dioxide comes off of it. 2 ATP are again produced. And the most important part at the end of it, right before it gets started again, NADH is made, which is what you need for the next step of the oxidative phosphorylation. You want to explain that?